Good evening, everyone. Today, our Department of Computer Science and Electronics, Ramakrishna Mission Vidyamandira, going to start the departmental student colloquium lecture. Today is the first day of this colloquium lecture. The purpose of this student colloquium lecture is to arrange an informal meeting where different students deliver lectures on different topics at each meeting. Each presenter has to entertain questions based on their delivery. This certainly improves their explanation capability. This meeting will be of a dual nature, where an address is followed by a question-answer session. This kind of colloquium is meant to deal with current cutting-edge concerns through preparatory study followed by a discussion. It is an added benefit to use supporting materials such as poster, handouts, and graphics. They add to the effectiveness of this kind of event, and attendees will receive something as a takeaway. Today's speaker is Shree Mitra, a first semester student of MSc Computer Science and Machine Intelligence. He will be delivering a lecture on Basic of Blood Cell Segmentation and Introduction to Automated Blood Cell Segmentation under the lecture series of an overview of automated blood cell segmentation. Now, over to Shree Mitra.
Thank you, sir. Sir, is my screen visible?
Yeah, it's loading. Yeah, it's visible. Carry on.
Good evening, everyone. My name is Shree Mitra, and today I am going to present the day one lecture basics of cell segmentation techniques and an introduction to automated cell segmentation techniques, which is a part of my lecture series and overview of automated blood cell segmentation techniques. So before going dig deeper into the main content of today's lecture, let us first review the overall lecture series. So day one, basics of blood cell segmentation and introduction of automated cell segmentation. In this lecture, I will give you the very basics concepts of cell segmentation and how automation comes into play in this field. In day two, we are going to traverse some basic image processing techniques like thresholding, filtering. In thresholding, we are going to cover OSUS thresholding, binary thresholding, inverse binary, etc. In day three, an introduction to OpenCV and Keras for basic image processing techniques. In this lecture, I will give you the practical implementation of some basic image processing techniques using OpenCV and Keras. See, OpenCV is enough for basic image processing techniques, but I have visualized some more improvement when we use Keras for basic image processing techniques. It will improve our model. Then we will go through some thresholding techniques for blood cell segmentation, and in day 5 we see an advanced algorithm known as seed watershed algorithm for blood cell segmentation. In day 6, we are going to cover some pitfalls of the traditional methods for automated blood cell segmentation, and we are going to introduce some machine learning techniques for blood cell segmentation. In day 7, we are going to create a simple artificial neural network model using TensorFlow, and we are going to classify the digits, handwritten digits using MNIST dataset. And in day 8 we are going to introduce the concepts of convolutions and convolutional neural network. And then in day 9 we are going to create our first CNN model for object classification. Basically we are going to study the CIFAR-10 dataset. In day 10 we are going to implement a blood cell segmentation using UNet, which is a very famous deep learning model for which is basically built for blood cell segmentation, semantic segmentation. In day 11 we are going to see a practical implementation of watershed algorithm and UNet. By combining these two algorithms, we are going to use an instant segmentation of blood cells. And in day 12, we are going to review some state-of-the-art deep learning models like Mask-R CNN, UNet for multi-class segmentation, and generative adversarial networks. In day 13, we are concluding this lecture series. So, in today's today's topic is basics of blood cell segmentation and introduction to automated blood cell segmentation. So today's agenda is at first I am going to cover blood cell image acquisition, then blood cell segmentation, then importance of blood cell segmentation. After that, manual segmentation. After that, automation in blood cell segmentation, and after that, different approaches for automation for blood cell segmentation. So at first, we are now going to talk about the blood cell image acquisition. So it is very important to know that before going to imply any kind of image related analysis on blood cell images, it is very important to collect the images, and we have to know how the blood cell images are collected. So blood cell images has been collected by magnifying a small amount of blood sample in a glass slab with the help of the microscope. And each drop of blood contains approximately 5 million red blood cells, and it is also been observed that there are 1 to 200 red blood cells for one white blood cell. So we can see that there are a lot of red blood cells than a white blood cell in a particular blood cell microscopic blood cell image. So a raw blood cell image contains a huge amount of blood cells, and for this reason, conducting cell-related analysis in these kinds of microscopic cell images is not possible. For this reason, the microscopic blood cell images divided into many segments, and we are going to imply image-related analysis on one of the particular segments at a time. So here is a segment of a blood cell. Here is an image of a microscopic blood cell image is shown here. So now I'm going to talk about what is blood cell segmentation. So blood cell segmentation from a microscopic image can be defined as the process of splitting a microscopic image of a blood sample into various segments in which each segment represents a particular blood cell of the image. Now here I have shown a very naive procedure of blood cell segmentation using simple Python program. Now importance of blood cell segmentation. See, blood cell segmentation is a very important research field in hematology research. By using cell segmentation, scientists are able to analyze relevant biological features such as cell count, division, shape, etc. Cell segmentation gives an approximate morphological structure of the cell present in the image, by which we can predict some diseases like myelodysplastic syndrome, etc. Now I'm going to talk about the process of segmentation. So here is a pictorial representation of the process of segmentation is shown, where the process of segmentation is divided into two parts, manual segmentation and automated segmentation. Now let's talk about the manual segmentation part. In manual segmentation, basically the hematologists have segmented and counted the number of white blood cells from a slide smeared with peripheral blood or bone marrow, or for the purpose of diagnosis and streaming. The hematologist manually segments and counts hundreds of white blood cells with the help of a microscope, and then calculate the total occurrence of each type of cells. This is called the differential blood count, DBC. This task is consuming, complex, and tedious. Now, in this process, the recognition accuracy largely depend upon the subjective factors like experience and fatigue due to human tiredness. By increasing the demand of the segmentation process, the manual segmentation will become an inhuman task. Now, the increasing demand of segmentation process for hematology research brings the necessity of automation in this field. Now I'm going to show you all a demonstration of a manual segmentation. This is a very popular software known as Visual Geometry Group Image Annotator, which is designed by the Visual Geometry Group situated in Oxford University. So we are going to annotate as image. First select an image. This is basically a cell image, a microscopic image of erythroid and myeloid cells. There are mixed myeloid and erythroid cells. So I'm going to annotate this. First, let's zoom. So I have annotated a particular cell, and you can imagine how much time is required to annotate the full microscopic image cells. And if there are thousands of slides, then it will be kind of impossible. That is why the necessity of automation in this field is required. Now, I'm going to talk about the automation in blood cell segmentation. Now, big question is can we design an automated system for blood cell segmentation? The answer is yes, we can. Many researchers have given many methods to solve this problem. Automated cell segmentation is tough because there are many variations which can be found in the structure of the cells, like congested cytoplasm, overlapping nucleus, and many more. I have discussed some of the very basic approaches which are being used for automated blood cell segmentation. Like thresholding was one of the earliest methods implemented for image segmentation. In the case of blood cell segmentation, thresholding was followed by morphological operations. In most of the cases, thresholding is computationally cheap and fast. In this case, we can build a semi-automatic system because for every new blood cell image, we need to update the morphological operation. In this case, thresholding is basically used for foreground or background segmentation of a cell image using binary segmentation or a binary thresholding, inverse binary thresholding, Otsu's thresholding, and etc. Now, our different approaches, region-based approaches, work on a certain criteria of homogeneity, usually of same or similar brightness of colors. All the pixels that satisfies the homogeneity criterion were grouped together into a region. Now, in this lecture series, I am going to make a semi-automated blood cell segmentation system. And after that, we are going to see some machine learning based segmentation methods. And after that, we are going to see some deep learning based cell segmentation methods like UNet, Mask-R CNN, generative adversarial networks, and multi-class UNet, faster RCNN, etc. So this brings us to the end of this presentation. So thank you for attending this presentation. Thank you, sir, for giving me this opportunity.
Thank you, Shree Mitra, for your excellent explanation. It was indeed a nice demonstration of the basics of blood cell segmentation. We hope that in next successive lectures, we will cover a lot more. And now this session is open for question answer. So if you have any questions, you can ask to him directly. Question from students. Yes. Any questions from students?
No, sir.
Okay.
Can I ask some question?
Yes,
Actually,
yes,
it's
yes.
not questions, it's some suggestion for him, Mitra. Mr. Mitra.
Yes, sir.
So I want to suggest you something. That most of the time I found that you rate on line by line over there. Okay, many places I have found. But in the presentation, especially at the time of presentation, you should not do like that. You should keep some reference point only. Okay, so you should be more interactive, more better you prepare like that. You just prepare a separate note for that to giving your talk and the some hint point. And simultaneously I want to suggest you many things that many slide you will you have made that corresponding picture can be implement on that particular slide itself. You can understand, right? So
Yes,
we are
sir.
not expert on particularly this field, so we cannot ask from that field itself. Okay, so and apart from that, uh, what I understood that you are going to able to you are trying to make some database related to it, right? Am I right, Mr. Mitra? Over to you.
Yes, sir.
You want to make it some database regarding it. Okay, so that you must implement over there. Emphasize rather than other things. What kind of database? What are some chart, something that visible things you should mention on the slide. That's all. I think it's enough hint that I gave you. I guess you have understood, and you
Yes,
will
yes.
implement in your next slide.
Yes,
Next time. Okay. Thank you.
yes, sir.
Okay.
Sir, I
Thank
have
you.
one question.
Yes, yes.
From where do we collecting this blood cell data sets? Is there any libraries in Python?
There are many not many, some open source databases you can find in Kaggle. From there, you can download the blood cell image databases.
Okay.
Okay. Any other questions? Okay. So, Sri, please tell us what's the application of this blood cell segmentation?
Sir, basically, by doing this blood cell segmentation process, we can actually identify different kinds of cells present in a microscopic image, and then after counting that different kinds of cell, we can estimate what kind of blood related disease the patient can have.
Okay.
Sir, I have a question.
Yes.
Sir, at the time when you're training those model by using this type of data set, suppose by using VGG image annotator, we are first annotate those cells by hand. Suppose this cell is this kind of cells, and first time we have to classify those cells and feed those images to our neural network model. So, is there any method to automatically annotating the whole data set without any manpower?
Annotation itself a supervised process. Like
Okay.
to do annotation, um, we have to annotate each and every cell. Uh, this is not possible without a human intervention. The annotation itself is a supervised process. We can do totally automated techniques using deep learning and image processing techniques. Like I have told you that we are going to implement a seed watershed algorithm and UNet for instant segmentation. In that particular lecture, I will implement that one, fully automated cell segmentation without any human intervention.
Okay. 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 Okay.
Okay. One question from me, another question, that what are the challenges you have failed?
Sir, in which part?
That means whenever you were actually collecting all these images, okay, then you were trying to extract different kind of features. So is there any challenging tasks exist in terms of feature selection or feature engineering?
Yes, sir. Basically, the main challenge is the morphology of the cell. Different kind of morphology of cell we can find. Sometimes we can see the nucleus of two cells are merged together, and some granules appeared above the cytoplasm portion and congested cytoplasm. This kind of abnormalities creates challenges at the time of feature engineering.
So how will you eliminate all these kind of things? Certainly when some outliers is coming in your pictures, how do you handle these outliers?
Sir, actually, to eliminate this kind of challenges, at first we are going to implement some basic image processing techniques like morphological operations, like uh, many kinds of morphological operations, filtering, etc. And after that, we are going to feed that image on deep learning neural network. And I think after that, we will get a good, at least satisfactory result.
Okay. Okay. Thank you. Any other questions? Any other suggestions to him? Okay. So thank you once again. And now this brings the end of this session. And we will come back very soon with another new and interesting lecture. Till then, goodbye. Thank you everyone for joining with us.
Thank you.